Hello, guys. Let me hope that you are doing fine wherever you are watching this video from. Today, I just wanted us to focus on some of the food tests. This is actually the biological practice or practical biology, but it involves the having some understanding in chemistry, not some actually having an understanding in chemistry. So this is for the person who do both biology and chemistry. So we are getting this as as a laboratory notes. We are going to test for food, or we are going to do what we call food test. Actually, saying food tests, it's about testing for the nutrients. We have the carbohydrates, which are actually called sugars. The very common carbohydrate is starch, which is commonly seen in many kind of complex foods, and we have lipids, which stands for actually fatty materials like oils or fats in general. And another thing is about proteins and vitamin C as one of the vitamins which is commonly seen in the fruits. So today we are just finding out how you can test for starch, lipids, proteins, and vitamins. Actually, here we are going to choose one thing which will be standing for the whole group of things. For example, for vitamins, we'll be dealing with vitamin C, and for sugars, we'll be dealing with starch. Lipids, it's actually the general test for all lipids, and the probably for proteins is the same. So we have to first take the safety note. Food or drink used in science laboratories must never be consumed as they may be contaminated. When you're in the laboratory, the kind of food that you're using for testing are not for like dietary consumption, or are not actually for eating, because they might be contaminated by some of the chemical solutions which are present in the lab. So we have many ways to perform the food tests. So all we have to do is to follow all the steps that we are given to just perform the test and mix well the reagents and just wait for an observation. If you get an observation, you have to draw a certain conclusion saying that the kind of food that you are testing for, a vitamin, a lipid, a protein, or whatever is present or is absent according to the observation that you'll be seeing. So the first thing is getting or setting well your materials or the working of your testing, and taking well your reagents, and just perform the test as it is instructed. Then see or observe. After observation, you will draw a conclusion. So let's start by vitamin C. Actually, the vitamins are mainly seen in many kind of foods, but also mainly in fruits. So vitamin C is also known as arzobic acid, like a chemical name. A very chemical name of vitamin C is arzobic acid. It is water soluble. That's one of the characteristics of vitamin C, and it is naturally present in the variety of fruits and vegetables, as I was saying before, fruits and vegetables, and is also a dietary supplement. If you say dietary supplement is something which you can get as one, like you can get as one nutrient itself. You can go to the pharmacy and they give you vitamin C only, not getting it from the kind of food that you consume. You can get a vitamin C as a chemical itself. So that's what we call a dietary supplement. So it says include oranges, strawberries, capsicums, black currants, broccoli, Brussels sprouts, and many fruits and the vegetables. Vitamin C is also a reducing agent. Yes, these are the chemical. Actually, we are saying about the chemical characteristics because the kind of test we are going to use will include using chemical reactions. So we have what we call an indicator called DCPIP 026 dichlorphenol, dinophenol, DCPIP can be used to test for the presence of vitamin C. It means you will be adding the DCPIP in some solution that you think vitamin C is present, and you wait for the observation. So if the CPIP will change, is added in the solution where vitamin C is present, the color will change in color from blue to red in the presence of an acid, but loses its color loses its blue color in the presence of vitamin E or vitamin C. No, it means if the if there is a kind of an acid which is present, the color blue will be lost. So it will also lose the blue color if vitamin C is present. So let's see what will be happening here. So we need this kind of materials: food samples that contain vitamin C, ascorbic acid, 0.0 ascorbic acid, 0.1%. That is the percentage actually. DCPIP solution in a dropper bottle, test tubes, dropping pipette, deionized or distilled water. Distilled water, it means pure water without any mineral. So we will add 2 milliliter. Actually, these are 2 drops of the DCPIP solution to a test tube. If using a liquid such as orange juice, add it drop by drop to the CPIP solution in a test tube, mixing after each drop is added. It means you have to mix and adding the orange juice, for example, if it is a solution containing vitamin C that you have, and if adding, you will be just shaking or mixing very gently, and after that you will be adding dropwise, drop by drop. So when testing solid foods, a liquid sample should be prepared by crushing the food in some deionized distilled water and using only the liquid in the test procedure. It means if you have some of the foods which are solid, you have to crush them and mix them with distilled water to make a liquid solution that will be using it with the DCPIP solution for testing. Add the liquid sample drop by drop to the DCP solution and test tube. Mixing each drop is added. As we were saying it before, you will be adding a drop and just shaking very gently, mixing the whole mixture for allowing the change to come about, to come around. So the color will change from blue to red if the sample is acid. If the sample is acidic, the color will change from blue to red. Remember that we're using an indicator. So continue to add more of the test sample, and if the color of the DCPIP disappears, then it shows that vitamin C is present. It means many of the solutions where vitamin C is present, they're acidic. So the DCPIP actually, if you start with it, it is blue. So if you mix the DCPIP, oh sorry, my internet is not working well. So I'll be waiting for the picture to come up out. So if you're mixing the DCPIP with the solution that we're using for testing, the blue color will change to red to mean that there is an acid. And when you continue shaking, then the red color disappears. It means vitamin C is present. So the 0.1% ascorbic acid, which is actually vitamin C, is used as a positive control and the water sample as negative control. This activity can be extended to utilize a titration method to determine the amount of vitamin C present in a food sample. You can continue using titration to determine the sample or the vitamin C which is present in that sample. Actually, this is very known in chemistry. I think you've done the many works in titration, which is actually good. So um, there is also testing for proteins in food. there is many ways that you can test for proteins in food, or if a protein can be found in food. So the very common protein which is found, which is universally found in many animals. Actually, proteins are found in actually food product, animal food products. It means meat, milk, eggs, things like that. So gelatin, it's because the proteins are one of the materials which make body tissues. So gelatin is one of the protein which is derived from animal tissues such as skin and bone. Gelatin, yes, such as say, skin and bone. So the Burette test is used to detect the presence of peptide bonds in proteins. We can test for the if the peptide bonds are present in our solution. So if we find that if the test become positive, so we give the conclusion that the we give the conclusion that there is the presence of proteins because we found the peptide bonds there. Remember that in proteins we have peptide bonds. So it can be carried out in several ways. We can use addition of Bullet reagent or addition of sodium hydroxide and copper sulfate solutions to the solution that we are testing if gelatin or if the proteins are present. So if protein is present, the solution turns from blue to purple. The solution that we added in the container where we are testing from will turn from blue and changes to purple due to the complexing of copper 2 ions. Yes, if you are just referring to this one, due to the complexing of copper 2 ions with the peptide bonds in the protein sample. The more peptide copper complexes that are formed, the deeper the purple color will come along. So if you mix sodium hydroxide and copper sulfate in the solution where we think that the proteins are present, there will be the color will change of Burette reagent will change from blue and changes to purple, and that is because there will be a formation of complex bond between copper 2 ions and the peptide bonds. So the more you form those bonds, the more the purple color become deeper. So test strips, note the test strips can also be used to detect some proteins, for example, Arubistic or Uriscan. Gelatin solution freshly prepared is generally used in the school science laboratory for the positive control. The ionized water can be used as negative control. Positive control is just for allowing us to get the proteins if are available. The negative control will just dissolve out the kind of testing things we are having. So the materials that we'll be having is 1 gram of gelatin, Burette reagent of 2 with this concentration of 2 m sodium hydroxide and 1% of copper sulfate solution, 100 milliliter of distilled water, stirring rod for mixing, test tubes, electronic balance, spatula for measuring, and 1 milliliter pipette. So the preparation is a 1% weight over volume gelatin solution. Weight out 1 gram of gelatin powder. Add 100 milliliter of distilled water in a beaker. Warm to around 50 degrees Celsius and still to dissolve. Stirring is like mixing things up. So cool to room temperature before use. Then add the gelatin solution that you've made in a test tube. After adding that, add 1 milliliter of Burette solution, or add 0.5 milliliter of 2 m sodium hydroxide solution, followed by 0.5 milliliter dropwise of 1% copper sulfate solution. So you have to choose if you add Burette solution or if you add copper sulfate solution. For both methods, either if you used the first method by adding the Burette solution, or if you use the second method by adding sodium followed by copper sulfate solution, for both methods, mix and allow to stand for 5 minutes. If you either use the first method or the second method, you are always obliged to mix and wait for the solution to stand and for around 5 minutes. So then observe a color change from blue to purple, indicating the presence of full of protein. It means if the color changes from blue, actually you know that copper sulfate solution is have blue color. The copper sulfate solution have blue color. So if the blue color changes to purple, so it means there is presence of protein. So if it does not change, then it means there is no protein. So the burette solution also is blue. So when testing for food, prepare a liquid sample by crushing. Here is for things which are like solid. If you have solid things, you have to mix them with water to make the liquid samples, because testing requires that the kind of compounds that we're testing on be in liquid form. So testing for glucose. How will we test for glucose or a reducing sugar in foods? All monosaccharides and some disaccharides are reducing sugars. Some examples are glucose, fructose, and lactose. Actually, glucose, fructose, and lactose are monosaccharides. Remember the types of carbohydrates. So here we'll be needing a Benedict solution, which is used for testing for reducing sugars. The copper 2 ions present in Benedict solution are reduced by these sugars to an insoluble brick red copper iron oxide, which precipitates. The blue color will first turn red. These are actually observation. The blue color will first turn green, then yellow, and may finally form a brick red precipitate. It means if you add the Benedict solution, naturally Benedict solution is contained in with copper 2 ions. So those copper 2 ions, when reacted with reducing sugars, or when they are reduced by the sugars, glucose, fructose, or lactose, they turn their color. From yellow, from green, from being green to brick red. So it means the observation that you'll be having is that the blue color will change to green, then yellow, and then it turns to brick red precipitate. The amount of reducing sugars can be related to the amount of precipitate formed. You understand? If the precipitate is very many, or if the precipitate is too much, it means the reducing sugar is also in high concentration in the solution that you are testing for. So test strips can be used to test sorry for the presence of glucose, actually, you understand. So glucose solution freshly prepared is generally used in school science laboratory for a positive control. Actually, if you've prepared glucose freshly, you can use it for testing, and the deodorized water, the deionized water can be used as a negative control to reduce the amount of sugar which is present, or reduce the intensity of the observation. So the materials that we'll be needing is glucose powder, electronic balance for measuring, spatula for like picking up the amount of glucose to be tested, stirring rod for mixing, 1 milliliter pipette. So then, what's the test for glucose? Test strips such as Diastic, Cleanstick, Curastic are similar available from pharmacies. Actually, these are materials or things to use in testing. So you can go buy them, but for you, you'll be actually be provided with ordinary things to use them for testing. So then the Benedict solution in a dropper, but the method to which is the one that we'll be using, we add a Benedict solution in a dropper bottle, 1 milliliter deionized or distilled water, Bunsen burner, tripod, and goose. Goose is actually where we put our beaker when it's on Bunsen burner. Test tubes or 250 milliliter beaker, 400 milliliter beaker, half filled with tap water for use as water bath. You understand? So water, like taking the water just for removing or just for cleaning up the testing material that we had. So then so, how will we prepare the glucose solution? We take 1 gram of glucose powder and we add it 100 milliliter of deionized or distilled water in beaker. and we will still till there are no crystals in the beaker actually when the glucose will dissolve. Then uh, and on the method two, we will add 1 milliliter of the 1%, actually 1% for concentration, glucose solution to a test tube. Then we will add 1 milliliter of Benedict solution and mix. We take a test tube, then we mix inside or put there the Benedict solution, and we mix it up so that it can the everything is mixed up. The mixture is homogeneous, of course. Then place the test tube in a beaker containing boiling water. We will have a boiling water. Then we take a test tube and put it inside in the boiling water. So we will boil it gently for 2 minutes, and we'll be observing the color to change, and the precipitate will be formed. A brick red precipitate, remember, will be formed. Brick red precipitate is that red which looks like those of bricks, matafari. So the brick red precipitate indicates a positive result. So if we find the brick red precipitate, we will say that the glucose is present in this solution. So when testing for solid foods for the presence of reducing sugar, we will need to prepare the liquid solution of it. So we have to scratch it, and add it to the water gently, and we have to mix. Yes, we have to mix. So how do we test for starch in foods? How do we test for starch in what? In food. So starch is a complex carbohydrate which is found actually this starch is a polysaccharide. Is a polysaccharide. So it's a wide range of foods such as potatoes, rice, corn, pasta, and grains. Many foods contain starch. Actually, carbohydrate sources, they contain the starch. So it is a mixture of polysaccharides, arizo amylose and amylopectin, which vary in concentration depending on the type of starch that we have. So starch would be or will produce a colloidal solution in water, as it is not very soluble. Starch is not very soluble in water. Starch is not very soluble. It's like slightly soluble, not wholly. Iodine solution is used. We will use iodine solution for testing for starch. If starch is present, it will react with iodine solution to produce blue-black starch iodine complex. Blue-black starch iodine complex. So a 1% of starch solution is generally used in school science laboratory for post. Actually, this test is also dukora for school. Zikorerwa mu mashuri. Then, these are one of the materials that we'll be needing. We need 1 gram of starch. It can be a corn flour, ifu, potato flour, or rice flour, any kind of flour that you can find, which is produced from the carbohydrate source. And then 1 milliliter of distilled water, hot plate or Bunsen burner, tripod like a stand, and goose electronic balance spatula, 250 milliliter beaker, 100 milliliter measuring cylinder, stirring load, test tubes or spotting tray, plastic petri dish, 100 milliliter pipette, iodine solution. Um, Iodine solution, or a potassium iodide. Yes, you can even take potassium iodide in a dropper. We put that in a dropper bottle. So we will add 1 milliliter of the 1% weight of a volume starch solution to a test tube, and 2 to 3 drops of iodine solution, and a blue-black color indicates a blue-black color. If we get a blue-black color after adding the 2 or 3 drops of iodine solution, it means there is a presence of starch. It means there is formation of starch iodine core. Starch iodine complex is formed. So, alternatively, a few drops of starch solution can be added to a well-spotted tray of 2 drops. We can add iodine solution to starch solution, or we can add starch solution to iodine solution. Or we'll be having the same result if the starch is present. So, how do we test for how do we test for, sorry, how do we test for lipids? Actually, lipids are all things uh, which contain oils. Actually, lipids are oils. Oils, they can be from cheese, they can be from vegetable oil, everything that is insoluble in water, like totally insoluble in water but being soluble in some organic solvents. So lipids really consist of fats and oils that are soluble in organic solvents in ethanol but insoluble in water. We know that oils are not soluble in water. So lipids are made up of fatty acids and glycerol. Lipids are actually made of fatty acids and glycerol. There are several ways to detect the presence of lipids in the food sample. Commonly used are ethanol immersion test and grease spot test. We have grease spot test. Actually, this grease spot test, you can even do it at home. This is actually it's a simple test. You take a drop of oil and put it on a very bright paper and put that paper near a candle. And when you see that where you put the oil drop is being more transparent, then you will say that the lipids are the lipids are available or are present in that mixture that you put on the paper. And we have another kind of test which is called ethanol immersion test. Ethanol immersion test, sorry. So we will get a vegetable oil as a sample because it is very common, or it is easy to find it than other kind of lipids containing materials. So it is generally used in school science laboratory for positive control. And vegetable oil and food sample containing lipids like milk, cream, cheese, or yogurt. Then we will take in ethanol immersion tests. We will take ethanol, pastel pipette or dropper, test tubes, and distilled water. And if you want to do the grease spot test, you will bring a brown paper bag, strips. Yes, I said the paper. You will take a paper bag or a paper generally, and we take cotton buds, light sauce such as lamp, knife to cut solid pieces of food samples, everything like that. So then, in the first method, combine 20 drops of oil or liquid food samples with 2 milliliter of ethanol in test tube. Then mix them well, allow them to settle for 2 minutes to allow any lipid present to dissolve in the ethanol. Add 2 milliliter of water directly to this test tube and mix gently. If using a more solid food sample, first allow any particles to settle, then remove the clear liquid component into another test tube containing 2 milliliter of water and mix gently. Actually, you have to take the drops of oil and mix them with ethanol very well till they are mixed up. Then the solution will turn to cloudy white color if lipids are present. We will take to cloudy white color if lipids are present. So if you have ethanol, you can also get ethanol where you live. You can go to the beauty salon or the barber shop and take that alcohol, iriya alcohol bagusiga ku mutwe. Iyi alcohol rero uyifashe, if you take that alcohol and mix it with amavuta yo mu rugo, niba wafatira amavuta yo guteka, then ukamixinga, wenda ibipimo nibyo byaba ikibazo, wenda utahita ubimenya, ariko 2 ml ni 2 drops of course, ukamixinga, uzabona igikara gisa umweru ariko byenda gusa nk'ibicu. You can see it, yes, byenda gusa nk'ibicu. So rero, that is what we call the ethanol immersion test. Then kuri second method ariyo twita grease spot test for oil or liquid samples. apply a small amount to a cotton bud and swap directly onto the piece of brown paper. Urafata cotton like ipamba ukoze niba wenda ari nk'ibintu biri solid, niba ari nk'amavuta ari solid uyakozemo, then ufata ipamba ushishe ku rupapuro nyine uzirimanganye ku rupapuro, if you can take it that way. So then we will allow it to dry for around 5 minutes. Turabitegereza nk'iminota 5. Then hold the piece of paper bag up to a light. Urafata nyine urupapuro urwegereza urumuri cyangwa ururebeshe ku rumuri, and look for a translucent spot that will indicate the lipids are present. Nubona aho hantu habonerana kurusha ku zindi mpande, tuzahita tuvuga yuko the lipid iri present. Just ibyo ngibyo byonyine. Yes, ndavuga ngo aho hantu nyine iyo spot n'ubu actually uraza kubona ko nyine no mu buzima busanzwe ari byo nyine wirirwamo, ujya ubibona ahantu nyine ibiryo bifite amavuta nyine byagiye ku rupapuro, ubona ko nyine uramutse uhegereje urumuri haba habonerana cyane kurusha ku mpande. So that is one of the observation. Rero iriya test bayita grease spot test. So thank you so much. If you'll be having any, so it means if there are no translucent spot, then there is no lipids, of course, birumvikana. So if you may have any question, don't hesitate to just reach out to me in the DM, and I will help you to sort it out. Thank you so much for joining me in today. So continue doing too many or too much exercises about this topic. So I think everything was clear today. Thank you so much.